All right, we've been talking a lot about mitosis and how mitosis is a copy machine. But when you look around, you see that nobody really looks exactly the same. Even within your family, there's so many differences between everyone around you. So if mitosis makes exact copies, then how are we all different? And that's where meiosis comes in. Meiosis introduces a lot of variation into populations. We're going to talk about how that happens in this chapter. I also want you to remember mitosis was a copy machine, whereas meiosis is more like scissors. It's going to cut our chromosome numbers in half. So keep an eye out for that as well, and always make sure that you're keeping mitosis and meiosis separate. There's a lot of similarities, so I want you to notice those, but also notice how they're different. So far, mitosis is a copy machine. Meiosis is the scissors that's going to cut the chromosome number in half. Okay, so let's look at this, how mitosis and meiosis come into the life cycle of an organism. Let's look at these zebras over here. We know zebras are diploid, just like we are. There's humans right here. They're diploid, meaning that they have two copies of every single chromosome. One copy from their mom and one copy from their dad. That makes them diploid. Now, if two diploid organisms combined diploid cells to make an offspring, that offspring would have 4 copies of every chromosome. And we know that's not what happens. That's not viable. That's not something that occurs. So that number, that diploid number, the one from mom, one from dad, needs to be cut in half to make a sex cell. Now we call sex cells, which are eggs and sperm, we call them gametes. There's the term right here, gametes. So gametes are sex cells. In order to make those sex cells, we have to undergo a process called meiosis. So meiosis is going to cut the diploid number, the 2N, one copy from mom, one copy from dad, and cut it in half so that we have a haploid gamete. So only one copy of every chromosome, only one N. And that's going to be your sperm and your egg. That's the only place in your body that has haploid cells, your sperm and your egg. Well, sperms and eggs. So once a sperm and egg come into contact, that's called fertilization. At fertilization, that's where we go from 2 haploid cells back into a diploid cell, because haploid plus haploid will equal diploid. The diploid cell here is called a zygote, and that zygote is somehow going to grow into a diploid organism, a diploid baby and then adult. And that happens through the process of mitosis. That's where we're going to make copies. We're going to make a whole bunch of copies of those diploid cells to make a larger cell. So mitosis is important for growth, development, and repair. You get a scratch or a sore, it's going to heal over with mitosis. You're going from a zygote to a full-blown zebra, that's going to be mitosis. But then meiosis is going to be to produce sex cells. So only for sexual reproduction is meiosis used. Okay? We're going to have to go back and look at some vocabulary terms for a second. We can do it right here. Actually, let's go to this. This one's better. Okay, so what I have here are 2 different chromosomes. But the important thing about these chromosomes is that they're about the same size. They contain the same alleles in the same places, only one's from the mom and one's from the dad. So that makes these 2 here homologous chromosomes. They're the same chromosome, but one from mom, one from dad. Homologous chromosome, that word right there. Homo means same. We've had that word before. Homologous chromosomes. Now, after replication that happens during interphase is where this chromosome is actually going to be replicated, going to make another copy. And then this chromosome is going to make another copy of itself as well. So after replication, what these 2 homologous chromosomes look like is this right here. And they're still called homologous chromosomes. There's still only 2 chromosomes there. Because as long as they're attached at the centromere, they're still considered one chromosome. So there's 2 chromosomes here. But these now are sister chromatids. So the copies These replicated copies here are called sister chromatids. They're like kids holding hands, and they hold hands at the centromere, which you've had that term before for mitosis, but I will show you that here in a minute. Okay, so that's vocabulary words that you need to keep in mind. Homologous pairs. Alleles are the different versions of a gene. And sister chromatids are held together at the centromere after replication. Okay. So what happens in meiosis, and how is it like scissors? Well, meiosis goes through very similar process as mitosis did, where we have prophase, metaphase, anaphase, telophase, just like before. Only we do it twice in meiosis. And there's a few other differences too that we're going to look at. So meiosis 1, some of the important key points to watch for is that there's a lot of recombination occurring in meiosis 1. I'm going to show you a slide on the different ways that variation comes into play here. And then we're actually going to separate the homologous pairs. Remember the one from mom and one from dad? And here's one from mom and one from dad. After meiosis 1, they're going to separate those homologous pairs. One into each of the new cells, the daughter cells. So what that does is leave, after meiosis 1, these cells are now haploid. There's only one copy of each chromosome. Whereas we started out with one from mom, one from dad, now you only have one of those copies in each cell. So we went from diploid to haploid in meiosis 1. Okay, and that left us with two daughter cells. Now in meiosis 2, we're going to have the sister chromatids separate. So now those centromeres are going to dissolve and the chromatids are going to separate, just like in mitosis. That's what happened in mitosis. And then we end up with 4 daughter cells, and they're all distinct. They're all different, and they're all haploid. So we are going to talk about how that comes about. I'm going to skip a lot of this here. Okay. So here's what I want to talk about. Oh, here, you know what? Where'd it go? Okay. Okay, so remember from mitosis how in prophase you have the X's that are the sister chromatids held together by centromeres. Well, in prophase 1 for meiosis 1, what happens is that those X's actually line up as homologous pairs. So they're not going to line up in single file, they're going to line up in pairs like this. So you have one from mom, one from dad of chromosome 1. One from mom, one from dad of chromosome 2. One from mom, one from dad of chromosome 3. Now when they line up like that, they actually touch. This one doesn't show them touching, but they do, right here in the middle, kind of like they're holding hands and feet. And when they touch, it looks like this. They do something called crossing over, and this occurs in prophase 1. They're called tetrads. They form a tetrad, like tetra. When tetrads form, crossing over happens, and that means that, kind of like if you guys have ever played with Play-Doh, we do a lot of Play-Doh around here, and if you put two colors on top of each other, some of the color from one goes to the other and sticks. So like if I have red and yellow Play-Doh and I put them on top of each other, I might have a little bit of red on my yellow. That's what happens. It's the same idea here for crossing over. So part of the genes from the dad chromosome, the chromosome from the dad, sticks to part of the chromosomes from the mom, and they kind of swap a little bit of their DNA there. So you can see this one now has some mom, some dad, some mom, or this one's all mom, and this one has some dad, some mom, some dad, and all dad. So each one of these is now different. None of them are the same because the middle ones here have swapped some of their DNA. So that's a huge way that variation comes into play during crossing over, and this happens in, I don't have that written here. So crossing over is in prophase 1. We say, here, I can put it right here. Prophase 1. Okay, Now, independent assortment is another way that variation occurs within the cell. Independent assortment happens during metaphase 1. What happens here is when the pairs line up right down the middle during metaphase, they may line up where all the moms are on one side and all the dads are on the other. Or they could line up with mom on this side for 2 of them and then dad. Or 2 moms of different types of dad. Each one of these lined up differently. There's still going to be chromosome 1 next to each other, chromosome 2 next to each other, chromosome 3, but whether mom's on the right or the left is different on each one for different chromosomes. So that we have a whole lot of different combinations that can occur here. And this happens in metaphase 1 when they line up down that middle, metaphase middle. So we have the tetrads forming in prophase 1 where the pairs come together, the homologous pairs, and they hold hands in the middle. And we have independent assortment happening with metaphase 1 and how they line up. So that's where the variation comes from in meiosis and why none of us look exactly like any of our family. Maybe a little bit, but not exactly. And what that leads to is that when a male gamete and a female gamete come together for fertilization, here's how many different combinations can occur. Check this number out. Let's see, we got hundreds, thousands, millions, billions, 70 trillion, 368 billion, 744 million thousand chromosomally different zygotes are possible. And that's if crossing over only occurs once, we have this ginormous number possible. That's an insane number. That's with crossing over. This ginormous number of different combinations of zygotes are possible. But crossing over doesn't happen just once. It occurs several times on each chromosome. So the number's even bigger. It's an astronomical number of different possibilities that can occur between the fertilization of male and female gametes, even within the same family. So that's pretty insane to think about. I don't have much time left here. I'm going to tell you about how this is different from asexual reproduction. So in asexual reproduction, this is mitosis. For example, a starfish, if you lose a little piece of a starfish, did you know the whole entire rest of the starfish body will grow back? Sometimes you can find those laying around on the beach. You'll see like a giant arm of a starfish and then this little tiny rest of its body. That is a whole new starfish growing from a piece of a starfish. And that's going to be an exact copy. That's just growth of the same cells. That's diploid to diploid. That's mitosis. Mitosis is diploid cell making more diploid cells, no recombination, no crossing over, no independent assortment. Whereas meiosis, that's where we have a diploid cell that goes into different haploid cells. And those haploid cells have lots of variation and lots of combinations and have to be fertilized to make an individual. So keep those separate. Asexual reproduction is mitosis, and this is a word here, asexual, meaning without sex. Asexual reproduction, that's mitosis. Whereas sexual reproduction is meiosis, is going to be involved in making those gametes. Okay, I'm going to end this video here, and then I will look at, real quickly, we can go through a few of the phases of meiosis. I'm not going to go through that too much because I have a bunch of activities for you guys to do on those. You can look at the different steps, but I do need you to know that you will need to be able to tell the difference between prophase 1, prophase 2, metaphase 1, metaphase 2, all of these different steps from a picture. Okay, if you see crossing over has occurred, so you see some of the mom on the dad and some of the dad on the mom, see how they're different colors, then you know that that must be meiosis occurring. If you see tetrads where you have them touching like that, that's a tetrad, that's definitely meiosis. Because mitosis, they line up single file, whereas meiosis, they have tetrads where they're paired. All right, I'm going to end this one and I will do one more for you guys, fill in some of those other slides.